So a continuation of what we were just talking about, I'd like to illustrate the delta G equals delta H minus T delta S with the following equation, or with the following word problem. For the reaction, reactant A is in equilibrium to reaction B, or reactant product B, I'm sorry, at 298 Kelvin. The change in enthalpy is negative 7 kilojoules per mole. I'm sorry about the word wrap there, but that's a negative 7 kilojoules per mole. And the change in entropy is negative 25 joules per Kelvin mole. Okay, is this reaction spontaneous? Or, obviously not written here, but implied, is it non-spontaneous? Okay, so to complete this question and answer this question, what you have to do is look at our equation, delta G equals delta H minus T delta S, and, well, plug and chug. So our delta H is negative 7 kilojoules per mole. So you need to convert that to negative 7,000 joules per mole. Now, T delta S, well, T is 298 Kelvin. Those are in the proper units, because if you look at our units for entropy, it's negative 25 joules per Kelvin mole. So negative 25 times 298 is negative 7,004, or sorry, is, since it's minus T delta S, we've got to come up with this negative value, which subtracting negative becomes positive. So negative 7,000 joules per mole plus 7,450 equals delta G of positive 450 joules per mole. In which case, is this a spontaneous or a non-spontaneous reaction? Our delta G here is going to be positive, and therefore it is a non-spontaneous reaction. Okay, so a follow-up to that would be, should the temperature be increased or decreased to make this reaction spontaneous? Well, let's think about this. Negative 298, if it is going to result in a positive value, what we need to do is get this 7,450 to be lower. So what we would want to do is do this at a lower temperature. For instance, if you plugged in something like rather than 298 Kelvin, the temperature were 250 to, again, I haven't plugged in my tablet for writing, positive 250 degrees. What you would do is 298 times 25 gets us that 7450. 250 times 25 is going to be 6250. So we're talking about negative 7,000 joules per mole plus 6250, which would mean effectively negative 750 joules per mole. So that would give you a negative delta G, therefore a reaction that is spontaneous. So this direct question, should the temperature be increased or decreased? Well, you'd want to decrease the temperature. Note that it doesn't ask, this specific question does not ask you for, well, at what temperature would it be a spontaneous reaction? But keep that in mind. You have your delta H and your delta S values, so you could come up with that T equals delta H over delta S to figure out the temperature at which anything below that, the reaction would be spontaneous. And think about that as what I think is a pretty good test question. At what temperature? So this is asking very generally, but you'll see questions that are asking more specifically. All right, moving on. Now, delta G is going to specify the direction of reaction, not the rate of the reaction. So if we look at this reaction on the left-hand side, right here, and then we look at our products right here, what we're going to see is a couple of things about this energy diagram. Our y-axis is our energy. Our x-axis is the reaction coordinate. So what we have here are two lines. Our red line shows the reaction taking place without the presence of an enzyme. And notice our red line shows us without the enzyme, our blue enzyme shows with an enzyme present. Now, here in the middle, right here, one double dagger, that's going to show us the activation energy that we have to reach prior to getting to our products. So as soon as we can get up here, well then everything kind of tumbles down. Now, what we are seeing with our enzyme is that our enzyme is going to basically lower our activation energy. So just as an inorganic catalyst lowers our activation energy, our enzyme lowers the activation energy. Now, sorry, I had like a little gnat or a fly flying near my face. That's our activation energy difference, basically. Now, the difference between our reactants and our products is going to display the overall energy that's released during our reaction. This activation energy will allow us or enable us to determine the kinetic rate of reaction. Now, our enzymes will lower the barrier, just as I was saying. They will lower that barrier, that activation energy barrier. So an enzyme basically makes it easier to catalyze our reaction. Now, our relationship between the free energy, our delta G, and our KEQ, our equilibrium constant, can be displayed as follows. Delta G is equal to delta H minus T delta S. Delta G is also equal to negative RT times the natural log of KEQ. Okay, our R is going to be 8.314 joules per mole Kelvin. That's the units that I'm going to stick with. You could also look at this in terms of kilocalories, in which case it would be 1.9872 calories per mole Kelvin, but I'm going to stick with joules per mole Kelvin because it just requires you to convert fewer units. Our temperature is going to be measured in Kelvin, so make sure you don't get that twisted in temperature Celsius versus Kelvin. Okay, so that right there is one of the things on which we're living with, focusing on. Now, delta G is going to depend on the concentrations of our reactants because of the fact that our equilibrium constant depends on concentrations, because our equilibrium constant, well, it's the equilibrium constant. So concentration of products divided by concentration of reactants. So another way that you can look at a reaction is to look at it in the context of what's known as standard state, where our standard conditions are in effect. 25 degrees Celsius and 1 atmosphere pressure, and each of the reactants starts with a concentration, or each of the reactants and products starts with a concentration of 1 molar. So that is where our standard equilibrium concentration is going to be derived. So what that means is that delta G naught, or that KEQ naught that you're seeing right here, those values might differ slightly from a delta G because of the fact that these are starting at 1 molar concentration. Now, so what that means is basically we've got these two to consider, where delta G is from in any condition, delta G naught is our specific conditions, delta G naught prime, this last variable, is one other to consider. And that is where the concentration, or sorry, the pH of the environment in which reaction is taking place is at 7, The concentration of water is 55 molar, or 55.5 molar. So those are two kind of caveats, or two qualifiers, that delta G naught, delta G naught prime, all conditions which may be which may result in delta G values that are not necessarily the same. So that delta G naught at standard concentrations, you would look at something like this. Delta G is equal to delta G naught plus RT times the natural log of effectively our KEQ, our equilibrium concentration at your not standard conditions. Now, one way that you can look at making this sort of calculation, or working this sort of problem, would be calculate the delta G naught prime for the reaction A plus B yields C plus D at 25 degrees Celsius. So you're provided with a temperature. when the equilibrium concentrations are A equals 10 micromolar, B equals 15 micromolar, C equals 3 micromolar, and D equals 5 micromolar. So we want to go ahead and approach this as delta G is equal to, or delta G naught, our non-standard condition, is equal to negative RT times the natural log of RKEQ. So we have our equilibrium concentrations, as I described earlier, and we can set this up to be this. Delta G naught prime is equal to negative RT times the natural log of our concentrations of our products divided by our concentrations of our reactants. Delta G naught prime is equal to negative 8.3 joules per Kelvin mole times our temperature in Kelvin, which is 298, times the natural log of our products divided by our reactants, which would result in negative 2473 or 2473. I don't know, I didn't say that number properly, times the natural log of 0.1. That effectively, it's 15 divided by 150, therefore 0.1 is where that number is coming from. So that's that right there. So the natural log of 0.1, which results in a value of positive 5.7 kilojoules per mole. Okay, so think about what that means. Our delta G naught prime, so the free energy change under non-standard conditions, well, is it positive or negative? This is going to be positive. Therefore, is this going to be an endergonic or an exergonic reaction? Is it favorable or unfavorable? What this shows is a delta G that is positive. Therefore, it is a endergonic reaction. Now, that's one of the things that I know people have a tendency to get mixed up on. They see delta G naught prime, and they say, oh, well, I know if delta G is positive, delta G is negative. I know if it's a spontaneous or non-spontaneous reaction. Oh, but then whenever I throw the naught and naught prime on there, does that change anything? It does not change anything. So the same rules apply, the same idea applies. The naught and the naught prime, all that these things are doing are saying something about the conditions. So it's just saying that it's not taking place at it's just providing you with a little bit more context for the reaction. It does not change the delta G being negative being a favorable reaction, or the positive being an unfavorable reaction. So keep that straight and narrow and everything like that. Okay, so one common reaction that you will see in this class as well as in a metabolism class is you'll know about ATP hydrolysis. So you maybe know a little bit about this from like a cell biology class, but this molecule right here is ATP, adenosine triphosphate. And some of those things might be like, oh yeah, I've got a little bit of context for that. The first of all is 1, 2, 3 phosphates. Okay, this molecule right here is adenosine. So we've got it's an adenosine triphosphate. Adenosine triphosphate is hydrolyzed to make adenosine diphosphate and inorganic phosphate. The delta G naught prime for that, so the free energy change for that reaction under non-standard conditions, is negative 7.3 kilocalories per mole. So units here are important. This is kilocalories per mole. Okay, this is overall though, independent of the units. What's the sign of that delta G? It's negative. Therefore, is it favorable or unfavorable? It's a favorable reaction. This is a favorable reaction because electrostatic repulsion amongst those phosphate groups is present. So to basically, they don't like being grouped with one another. And so if you can kind of throw one off, that's a positive thing, or that's a favorable thing. We've got resonance forms of our product, our inorganic phosphate, the Pi molecule. We've got cratic entropy, so we have an increase in the number of system components. And we've also got solvation, ionization, and release of a proton in this process. Now, a question from that is, if ATP hydrolysis is favorable and it's spontaneous, how can biological systems use ATP to store energy? So you have this molecule that I guess you could kind of phrase it as it breaks down easily without any sort of outside influence. Why would you want that as an energy storage molecule? Well, so what this slide shows is some of the information that we knew from previous, where our delta G naught prime is negative 7.3 kilocalories per mole. Okay, our R value is 1.9872 cals per mole Kelvin, and our temperature is 298 Kelvin. Now, our concentration for ATP, ADP, and PI is presented specifically in the context of human hepatocyte cells. Okay, so if you take into consideration the delta G naught prime for this, as well as the concentration of our equilibrium concentrations of ADP, PI, and ATP, well then the delta G for this reaction is negative 11 kilocalories per mole. So if you compare that, we have equilibrium concentration at standard conditions was, or sorry, at non-standard condition was negative 7.3 kilocalories per mole. Then when you have equilibrium conditions, given the equilibrium concentration of our reactant and our products, well then that actually makes this reaction more favorable. It goes from negative 7.3 kilocalories per mole under non-standard conditions to negative 11 kilocalories per mole. So this reaction is arguably more favorable in a human hepatocyte. So you might be thinking, well, the molecule will break down easily, but it will break down even more easily, I guess you could phrase it that way, or more favorably in a human cell. Now, how does that kind of play into the value of this molecule in a metabolic sense? Well, coupling of reactions is how. So what cells do is they couple, or specifically in this case, we couple ATP hydrolysis and group transfer to an unfavorable reaction. So to illustrate this, if you go on to take metabolism, you will see this over and over and over again. But what is involved in glucose breakdown, or glucose breakdown, glucose catabolism, the process known as glycolysis, is the first step is a transfer of glucose or inorganic phosphate onto glucose to make glucose 6-phosphate. Now the delta G naught prime for that is 3.3 kilocalories per mole. Now catabolism, I've already kind of implied this, but it's a set of metabolic pathways that break down complex molecules into smaller units to release energy. Now, energy coupling is taking essentially an exergonic and an anorganic reaction, combining them with one another to make the combination favorable. So we have ATP, which is a high energy molecule, which reacts directly with a molecule that basically is a lower energy, or a molecule that needs activation. So to illustrate that, on the left-hand side of this, you have reaction number 1, the phosphorylation of glucose to make glucose 6-phosphate. The reactants are at a lower energy than the products. Okay. Reaction number 2, ATP going to ADP and PI. We've got a high energy molecule going to a low energy molecule. Now, reaction number 3 is the combination of those two. Glucose plus ATP yields glucose 6-phosphate and ADP. So what this shows is it's almost like Hess's law when you think back to Gen Chem 1, where we took partial reactions, combine them to make one overall favorable reaction. So delta G3 is equal to delta G1 and delta G number 2. So we've got two reactions that they pair up to make one reaction favorable. So energy coupling is a key component of any sort of metabolic pathway. We will not get too far into metabolic pathways in this course. That's really reserved for a metabolism class. But to sort of sum them up, essentially what you have is a series of related enzymatically catalyzed reactions. So if you look at this display right here, it shows reactant A ultimately ending up as product F. And in that step, we have a series of enzymes that will produce intermediates that are then used for subsequent steps. So, though there are two pathways that I think are important for you to understand, a metabolic pathway is this one, whereas a signal transduction pathway is one in which you're transmitting information rather than developing a product of sorts. So I always kind of think about it as a metabolic pathway is much like or it's akin to like an assembly line, whereas a signal transduction pathway is a little bit more like the if you were in elementary school and ever played the game telephone, where one person gets a piece of information, tells it to the next person, tells it to the next person, tells it to the next person, and then the last person will relay that message. Unlike the game telephone, signal transduction pathways in in living systems don't have this sort of deterioration of the message and loss of the message along the way. So yeah, there's a fundamental difference. Now, pathways are controlled, whether they're signal transduction or metabolic pathways. They're going to be controlled um, in a couple of unique ways. For instance, here what we have is the pathway that takes the amino acid, or the metabolic pathway that takes the amino acid threonine through some level of catabolism. And one of the intermediates in that pathway is the also happens to be an amino acid, isoleucine. Now, isoleucine acts as a negative feedback regulator, and so it's going to block enzyme number 1, the first one that acts on the amino acid threonine. So what that means is basically like that isoleucine is a trigger to say, stop, we've got enough, we'll proceed, or stop, we've got enough, we need to kind of, handle what has been produced before we go on to produce any more. All right, so this concludes the physical steps or the physical foundations for biochemistry, and next you're going to be talking about, or I'm going to be talking about the evolutionary and genetic foundations, which are not going to be extremely extensive at any rate. All right, well, have a good one.